All right. Well, thank you for inviting me to speak at this forum. And I'm very happy to tell you about our adventures in genome writing, as we like to call it. I'm involved with a bunch of companies and institutes. As you can see here, some of them pay me money, so I'm required to reveal this to you. And I thought I would start with something that's already been mentioned, which is that DNAs in nature occupy quite an impressive scale there. You can see orders of magnitude. And let's see here. Okay. So here's the longest oligonucleotide that can be synthesized today. Here is you heard about the AAV vector before. That's about 5 kilobases. There are some other viruses that might go as high as 100 or more. The average human protein coding gene, 62,000 base pairs. The longest synthetic chromosome that we've made in our lab is 1.4 megabases. We're very proud of that. But as I'll show you in a moment, we kind of have a long way to go in synthesis when we compare to things like human genes, longest human genes, human chromosomes, and the world record holder, Australian lungfish, has one chromosome that's about twice as big as the entire human genome. But what I really want to do now is I want to emphasize something, which is that this is a log scale. Okay, and now you guys look at these all the time, but I just want to put this in context. So if this represents the whole human genome here, okay, 3 gigabases, this is 370 feet of strength, okay? So it would go from one end to the other end of a football field and still have another 40 feet. And now I want to compare that to my other favorite organism, which is yeast. That's the yeast genome, okay? That's a humongous difference. These are two eukaryotic cells. They have a nucleus. They package the DNA in nucleosomes, like you heard before, and they're condensed into chromosomes. But vastly different genomes and vastly different biology. And I actually put them on the diagram. So here's my first favorite organism, Saccharomyces cerevisiae, weighing in at around 12 to 14 megabases. And here's my second favorite organism and one of my favorite humans at about 3 gigabases. So that's Karen, in case you don't recognize her. Okay, so with that as an introduction, now I'm going to talk about what's at the top of the slide here, which is what we affectionately refer to as big DNA, which is in the 100,000 and up range. So, and I'll explain this in the context of the dark matter project. So this difference in scale here. Oh, I forgot to mention a really important point, actually, which is that by comparison, the HIV genome, okay, which is a virus, that 10,000 base pairs, that would be 35 microns long, okay, which is the smallest object a human eye can see. An average human gene, 62 kb, would be 1/16 of an inch. Okay, and there's 20,000 of those packed into this guy. 20,000, 1/16 of an inch. So there's a huge amount of empty space in here. Some of it's introns that you heard about before, and a lot of it is the space between genes. And then the average yeast gene, the yeast genes mostly don't have introns, they're very tightly packed, would be 5 microns, or the thickness of one strand of spider silk, which you can't even see with your eye. Okay, so now I'm bringing back the yeast here, and I'm telling you about the dark matter project, because what this means, if you put everything I said together, is that the human protein coding regions are these tiny little specks floating on a sea of dark matter, okay? And we want to understand that, and we're using big DNAs to do that. And so we developed this workflow where we go on a computer and we design a big DNA. We're interested in some gene or some region that controls a gene. We build it inside a yeast cell. I talk a little bit about that. And then we can take it out of the yeast cell and into a mouse embryonic stem cell. We do that in a couple of stages. I won't go into a lot of detail on this, but the remarkable thing about these embryonic stem cells is we like to sometimes call them the yeast of the mammalian world, because we can take this single cell and we can put it into a blastocyst, and that will produce an animal that has exactly the genotype of whatever engineering we did in these embryonic stem cells in culture. And that creates an animal that we sometimes refer to a genetically rewritten and tailored genetically engineered mouse model, or Great GEM for short. And then we can study these animals, and then we can go back and go around this circle several times if we like. So to make all of this easier, faster, cheaper, because this is not currently an easy thing to do, we are very fortunate to have a grant from the National Human Genome Research Institute to develop what we call the Assemblatron, and you're going to hear a lot more from that later this week, where we're trying to automate the process of the big DNA assembly in the yeast cells. And this, the vision here is that hundreds to thousands of 100 kb constructs in research could become routine. And we're hoping to partner with a robotics company to actually turn this into an instrument. And so the technology behind it is that if you want to build 100,000 base pairs, it's going to have some repeats in it, and those repeats can misassemble, because the way you put DNAs together is that like goes with like in a homologous recombination reaction, and if you have two red sequences, they can misassemble. So, and we always start with smaller pieces and build them up into larger pieces. And so there's kind of a segmentron that will basically assure that we don't assemble with overlaps in the repeated regions. Rather, we will segment in such a way that the repeats are hidden in the middle. And so anyway, that's one step. We also have a special cloning system that is going to enable us to automate these steps, and also have made improvements in the transformation that make it robot friendly. So basically a bunch of technology to make it go faster, cheaper, bigger. And so our original goal was to go 10 times as fast as we could before we got the grant. I'm happy to say that right now it's looking like we can do 96 times as fast, and we still have a year and a half to go. So very excited about that. And we're actually starting to think about turning this sort of monster machine here into a desktop machine that would allow a laboratory to make 4 to 40 megabases of DNA a month. And so Antonio is going to talk more about this later this week. So meanwhile, what are we building? We're still, in the slow lane, relatively speaking. So we've made about 40 or 50 of these mega DNAs, and we're using it to do two things. One is we want to understand what's all this dark matter doing to control gene expression. So we think of it kind of like a switchboard here. So I realize none of you have actually seen a switchboard, but the basic idea is here that these people are trying to connect person A with person B at the right time, and not make any mistakes and misconnect a call. So derangement of this kind of control would underlie nearly all common genetic disease. So the logic of these switchboards, here's actually a diagram of a volume control, and it's a complex thing relatively. And but it controls a simple output, which is the speaker on loud or soft or off. And just to give an example of the analogy to the human genome, so here I've zoomed in, there's a megabase of DNA there centered on a tiny blue gene here called MYC, which is an extremely important gene in cancer. It's an oncogene, and it's only 4,500 bases. And look what's lying to the left and to the right. There are no protein coding genes. There's some genes encoding gigantic RNA molecules, and there's all kinds of peaks here suggesting transcription factor binding. So this is what we mean by the dark matter, and we're trying to understand this one and many other ones. And then the second thing that we're doing is we're making these humanized animal models of both common and rare diseases. And these are going to play really important roles in drug discovery, and in particular for genomic therapies like gene therapy or antisense oligonucleotides, because this gene control is very complex, and we need to turn them not just on and off in the right time and place, but also set the volume control just so. And to safely deliver them, we'll probably need to test lots of variations on the payload. So this is sort of the justification for developing some of these animal models before we take therapies like this into people. So last, I'm going to tell you about a particular animal model that we've developed in the last couple of years called XDP. This is a rare disease, X-linked dystonia-parkinsonism, and every single case can be traced back to the island of Panay in the Philippines. It's a X-linked male-specific disease, and it has very specific symptomology that has actually a lot of similarity to Huntington's disease, but also has sort of echoes of Parkinsonism. Now, we believe that this is what's called a founder effect mutation. It happened once, probably to a guy, probably a very prolific guy in Panay, and every single descendant has the same haplotype in one position on the X chromosome. So this is the classic X chromosome inheritance through the female line, and only males are severely affected. And very characteristic problems in specific parts of the brain, the caudate nucleus and the putamen, and in some cases, very large, basically holes in the brain that are characteristic of this on death. Now, the causal genetic variant is, as Karen put it, a dog jumped into the middle of this intron, okay? So this is dog, all right? This is the dog that, came up at various places in the story. So there are 3,000 copies of a jumping gene or retrotransposon called SVA, and it's human specific. And one of them landed in this individual one time in this one spot in intron 32 of a gene called TAF1, which happens to be on the X chromosome. And work done by others, not us, showed that this is exon 32 and exon 33. Okay, so that those little blocks there are the tiny little snippets of coding region in this very important transcription factor called TAF1. It's an essential gene. And what they showed was that there's a lot of missplicing or termination occurring before you get to this SVA. And furthermore, when this SVA is snipped out, all of that goes away. So we came up with a way to develop a mouse embryonic stem cell model and a mouse model from that. So first step was we dropped a lock site right here, this red triangle, into one of the earlier introns in the gene. And then we dropped a landing pad downstream. And then we had to keep this green mouse gene active all the time, or the mouse cells would die, because the TAF1 is essential. Meanwhile, our colleague was building this set of constructs here, which have exon 25 through exon 38, and either with or without the SVA, and went to great pains to make sure that the protein coding was exactly the same. So we mouse-sized the exons of this gene. And then we delivered this construct either with or without the transposon. And then we could use the Cre recombinase to excise and make what we call a hybrid gene, and we call this conversion. Anyway, that's a lot of technical details, but just to show you, this was a non-trivial matter, and it absolutely required us to be able to do these big DNA manipulations efficiently. And at the end of the day, the really amazing thing is that this poor little guy here is the experiment, and on the right are the three controls. And I think you can tell that at the age of 26 days old, this mouse has severe problems moving around, and you'll see some evidence of tremor and other phenotypes reminiscent of the human disease. But amazingly, the humans that get this disease get it in their 30s and 40s, and typically die in their 50s. These mice all die by 8 weeks. So here you can see the males. It's a completely male specific, and they lose weight, and the females and the negative controls don't have any of those phenotypes. And then there are some characteristic movement deficits and behavior deficits characterized by our collaborators, Priya Prakash and Shane Littleow. And last but not least, a very, very obvious problem with the brain, again, very reminiscent of what's seen in the human disease. And finally, by dissecting out these regions of the brain, Wei-Min Zhang in my lab was able to show that indeed the presence of this transposon here leads to a deficit in the amount of full-length messenger RNA from this gene, which we believe is probably the fundamental problem. Also, what's very clear here is, were we able to simply snip out this one segment, we can restore the full expression of it. So this is why we think this could be a great model to test a potential gene therapy for this disorder. And with that, I will wrap up and give my huge thank you to my team in the lab at NYU, and also to thank NHGRI and the Collaborative Center for XDP for their support. Thank you. Yes, the bottom row is art made with genetically engineered yeast that express pigments of different colors.
Hey, Jeff. I have a question about TAF1. So it occurred to me that I want to be careful how I phrase this, because the field of genetics has made a lot of predictions based on EQTLs. And it occurred to me that maybe your approach might be a way to test it more so. And so I'm wondering if you could comment on that.
Yeah, so I didn't have time to talk about all the different kinds of things we're testing in the dark matter project, but one of the ones where we haven't quite come to the finish line yet involves looking at GWAS hits and EQTLs, where they come together, and whether we can dissect those GWAS hits to find the causative variants that underlie.
Do you think of those as the switches in your switchboard analogy?
Yeah, exactly. Okay. Exactly.
And then my second question is more of an evolutionary question. So TAF1 is an essential gene. Why would it be on the X chromosome? GATA1 is another essential gene. It's a hematopoietic master regulator. Why does it make sense to do that?
Yeah, I don't know what God was thinking. It's a good question. I think genes end up in different places essentially by accident. I mean, it still works most of the time, right? So, yeah, but it causes very specific types of disease when things go wrong.
How cheap does synthesis have to be before it becomes something that is going to be, you know, where every lab has a synthesis machine like they have a PCR machine now? And we haven't gotten that. That doesn't happen in the oligosynthesis world.
Yes, we've been waiting for that for a long time because you know, the price has gone down, and it has, as George Church really likes to point out, and he's totally right about this. The price per base in oligo pools is orders of magnitude lower than the price per base in individual oligos. So there are some new methods that have come out recently that seem like they might harness the ability to use those oligo pools to produce DNA at a much lower cost, and we're very excited about them, actually. So if we could start getting our 3,000 MERS, which is our preferred starting material, for a cost in the sub 1 cent range, preferably sub 1 cent, then it really changes the game.